now this is the time for the PHC 2020 award. And this award this year is décerned to Michael MANNS. Michael is not only a great hepatologist, he is a great internist, a great researcher, and a long career with many exceptional achievements in terms of research, education. And Michael is not only one of the best experts, maybe the best experts worldwide in autoimmune liver diseases, but he was also interested in viral hepatitis, hepatitis C. He participated and was a leader of many phase 3 clinical trials in hepatitis C. He also made a lot of research on end-stage liver disease, liver transplantation. And so I will say he is a complete hepatologist with clinical experience in all fields of hepatology. So he is certainly one of the strongest personalities in the last decades in Europe and in all the world in terms of production of research and clinical work. Michael is also a great friend, and I always appreciated in our collaboration in the past his friendly and very fair, very nice participation in different groups at EASL and also for building collaborations at the European level and at the international level. And Michael MANNS is actually president of Hannover Medical School in Germany. I don't know why the slides are going faster than me and faster than Michael. No, sorry. Okay. Anyway, he made his studies in Austria and then in Germany, and he was also a research associate in the Scripps Research Institute in California. He made a long, as you know, research mainly on autoimmune liver diseases and other liver diseases. And he was a chairman of the German network for hepatitis and also of the German Liver Foundation. He received so many awards. I tried to list that on slides. It's absolutely impossible. So I decided just to say I think he had all the awards possible for an hepatologist in his career. And also of importance, he produced so many articles, more than 1,000 articles in the major journals, including New England Journal of Medicine, Lancet, Nature, and the best journals. The age index, I didn't know that the age index could reach 159. So Michael is probably the winner of the age index. I have to check mine, but I will be extremely jealous. So this is Michael. This is the small one. You know, this is a child. It looks like Germany. Nice photo, and we can see that already Michael has curiosity and was a researcher. He's looking for something. Already he had the research, you know, gene, the DNA. And Michael, with his wife, Michael is a strong supporter of hepatology, but also a strong supporter of football, of soccer, as you can see. Michael, do you play football? Michael, where you are? Michael, do you play football? No, okay. You prefer to look at the TV. That's safer, I agree. And he has a strong production in hepatology. Also, he has a strong production in terms of family, beautiful family, father, grandfather, grand-grandfather, not yet. Okay. I was a little, I was thinking you are very fast. And this is a dry January. But Michael, on the left, is giving some champagne to, I think this is Professor Meyer-Zulmuschenfeld. I'm very sorry. I was very sorry to learn that he died a few months ago. He was a master. And you had the chance, Michael, to have a big master. He was one of the builders, of the founders of EASL, with Sheila Sherlock, Jean-Pierre Benamou, and other big hepatologists. So, Michael, thank you to be here. I'm very happy, as a great friend, to give you this award, this modest award, the PHC award, that you deserve for your long career. And your career is not finished, I guess. I have seen in your CV that you are starting your work, you have projects with your liver foundation. So maybe you will tell us what are your projects for the future. And Michael, you are the best to tell us now, in your lecture, about the future of autoimmune liver diseases. Thank you, Michael.
Yeah, Patrick, thank you very, very much. I feel very, very honored. I'm very pleased to receive this award, and at this time I want to thank everybody who traveled with me through this long-term journey, my family, my mentors, my collaborators in all the cities I worked with, in Mainz, in Berlin, in La Jolla, and in Hanover now for 28 years. Some of the data you have seen in previous talks from collaborators with myself. So now it's time to talk on the future of autoimmune liver disease. This is the journey where I started with Karl-Hermann Meyer-Zulmuschenfelde, and I give acknowledgement to the people that at present are working with me in the field. I also have some conflicts of interest. And there are three major autoimmune liver diseases. This is autoimmune hepatitis, tackling the main liver cell, the hepatocyte, primary biliary cholangitis, as we call it now, targeting the small bile duct, and primary sclerosing cholangitis, the medium-sized and larger bile ducts. Over the years, the numbers of patients being transplanted for autoimmune hepatitis stayed very stable, with around 4%. But we have seen changes, at least in Europe, for the cholestatic autoimmune liver diseases. There has been a sharp decline in primary biliary cirrhosis, maybe thanks to URSO, which was introduced in Paris to the therapy. And I remember one of my early hepatology papers, I worked on mitochondrial antibodies. In 1982, we published in PBC-specific radioimmunoassay, which turned out to be later identified targeting the PDH-E2 molecular target. And then we did studies with Groningen, because Groningen mainly transplanted PBC patients, which is now a rare indication. In PSC, it's increasing. In Hanover, it's now number 2 on the list of patients being transplanted for the liver. I want to start with primary biliary cholangitis. Ursodeoxycholic acid is the standard of care. And we all agree, and this is a cartoon from Ulrich Beuers, that ursodeoxycholic acid is not targeting the initial steps, the loss of immunological tolerance to biliary epithelial cells. And here we still have only limited knowledge. And it's targeting later stages on maybe the secondary toxic effects of bile acids. Nevertheless, we agree that 20 to 50% of patients respond to ursodeoxycholic acid, but not 50%. One of the dilemmas that we still lack endpoints, apart from histology, and another is that there's a slowly progressing disease. And when we look back, we must say how easy was hepatitis C.
And here you can see various endpoints for the response to ursodeoxycholic acid. In the focus is the alkaline phosphatase. And so, depending on the score system, it's 25 to 50% who fail to treatment of ursodeoxycholic acid. And what is also very important, this is one of the papers from Paris, that those who respond to ursodeoxycholic acid have a normal life expectancy, while those not responding, they have a need for novel therapies. And what is also important, that this also has an impact on the development of liver cancer. We all are aware that we have now an approved drug based on this phase 3 trial, obeticholic acid, and that, again, of those not responding or inadequately responding to urso, that again, only around 50% respond to this second-line therapy. And this is now a sub-analysis of this study published by Michael Trauner last year, looking those patients where OCA was put add-on to urso, and you see here the response, but again, the endpoint less than 1.6 upper limit of normal is not reached by all the patients. It's around 50%. And then we all were surprised by the bezafibrate study. Bezafibrate is off-patent, as we know, as an anti-lipid drug, and we see here that there's a significant decline in alkaline phosphatase for a drug which certainly is less costly than obeticholic acid and has less pruritus. What also was shown is that in the SCLOPE score, there is an improvement for those patients where beta is added to ursodeoxycholic acid. And we have heard that Intercept, who owns OCA, that they acquired the right to develop bezafibrate in combination with OCA for, and hopefully we have more responders for those failing to urso. This is an interesting oral presentation, which I have not seen as a full paper, was presented at EASL from the Leuven group, looking at those patients out of this OCA phase 3, the POISE trial, where then bezafibrate was added to OCA plus minus urso. And here you see that there was significant response, and this is forming the basis for the ongoing trials combining bezafibrate with OCA. However, what we are missing is an own arm of beta plus urso, as far as I know. After many years, with a little movement in the therapy of primary biliary cholangitis, we see now major drug developments. And due to limited time, I cannot go into all of this. You see that PPAR gamma, PPAR alpha gamma agonists are under development, FXR agonists, but also FGF19 agonists. Here you see preliminary data from seladelpavir, the PPAR delta agonist. And this is interesting, FGF19. You see a decline in alkaline phosphatase, which is the leading biochemical marker for treatment response. And this is of interest because it is targeting the route of bile acids in the intestinum, binding to the FXR receptor in the enterocyte, and then leading to FGF19 increased synthesis, which is then having an impact on CYP7A1, which then is limiting bile acid synthesis and may thus lead to a decline in the progression of cholestatic liver disease. And again, there are many other molecules now under development. I cannot go into detail. So in primary cholangitis, we will not stay with urso and OCA in the future. Now, primary sclerosing cholangitis. This is a disease with an undefined etiology, ill-defined pathogenesis. There is this high risk of malignancy, 15% bile duct cancer. There is 100% increased risk of pancreatic and colonic cancer. PSC patients with ulcerative colitis have double risk to develop colonic cancer than colitis alone. We have an absence of medical therapies, and liver transplantation remains the only life-saving intervention of patients, and therefore we call it the black box of hepatology.
What do we know about the pathophysiology of PSC? There have been various hypotheses. I come back to it later. But the most interesting in the past is on microbiota. And this publication comes from Nakamoto in Nature Microbiology just last year. There have been studies showing that there is a dysbiosis of the microbiota in the gut. But what is most interesting of this paper is that obviously Klebsiella pneumonia, together with Proteus mirabilis and E. gallinarum, these three together lead to a dysfunction of the GI barrier and then to an activation of Th17 lymphocytes in the liver, mediating the inflammation. And it's the three mixed bacteria that cooperatively promote the hepatobiliary disease progression through Th17 response, which in the future will give us new therapeutic targets. And everybody is doing these days microbiota research, but I think in PSC it looks very, very promising. So what's new about future therapies in clinical practice for PSC? And this is a cartoon I got from Michael Trauner from Vienna. You see that the hypotheses are targeting various molecules, either within the bile duct, within the intestinum, or according and based on David Adams' hypothesis, that we have leaky duct, that we have an activation of lymphocytes, which then are moving to the liver, and that gut-primed T cells mediate the inflammatory process of the bile duct epithelium, which then leads to obliterative fibrosis of bile ducts with all its consequences. And so therapies now are targeting the FXR receptor in the liver and in the enterocyte. They are targeting PPAR and ARR agonists to prevent progression of bile duct fibrosis. PSC may also be called, it's the cirrhosis of the biliary tree. And we have seen recent data on dysbiosis of the microbiota in the gut, but there have been some years ago already trials using antibiotics, vancomycin, minocycline, and metronidazole in order to modify the gut bacteria microbiota content, which may have an impact on cholestatic disease. And then there have been preliminary data looking at antibodies to integrin that may have an impact of the traffic of the gut-primed T lymphocytes to the liver. However, the study with vedolizumab, which is approved in inflammatory bowel disease, which initially got signals of a positive response also to those with concomitant PSC. However, they could not be confirmed, and therefore a planned trial was stopped. There's other drugs like Sancrivivoc, but very interesting, there's simtuzumab. It's an antibody, LOX2 antibody, targeting the fibrosis process. The study was negative, but the company behind that, Gilead, used the data of these prospective trials to come up with interesting data I will cite at the end of my talk. And there is Nordis oxycholic acid. It's an analog of erythritol oxycholic acid, which has an impact on the biliary circulation of bile acids, and I will show you some data. This is an FXR receptor agonist, cilofexor, for the treatment of PSC, and you see there is a benefit in alkaline phosphatase, but it's a rather limited benefit, so I think this target on its own will not bring the solution. Again, here now, microbiota, not basic research on pathophysiology in humans and mice. This is the first initial study published last year where a single, where by colonoscopy, FMT, so fecal microbiota transplantation, was used in PSC patient, and the microbiota came from a single donor, healthy donor, and you see decline in alkaline phosphatase and transaminase levels. Overall, this was safe, and it will be interesting to see future data. More closer to reality and closer to everyday praxis, and I'm one of the principal investigators. This is Nord also. This is data of a phase 2 study that was published 2 years ago.
or 3 years now ago. Michael Turner, senior author, and Peter Fickett from University of Graz. The patent is with the University of Graz, and Falk is developing it, and you see a dose-dependent decline in the alkaline phosphatase, which is also a marker in PSC, and phase 2 study, European-wide, is running and hopefully will be recruited very soon. Apart from transplantation on the one side and medical therapy on the other side, we should not forget about endoscopic intervention. Endoscopic intervention in dominant strictures is important, but is dangerous, has complication. One of them is cholangitis, one of them is pancreatitis. But there have been recent very interesting studies. One was one European-wide study led by Oslo and Amsterdam showing that balloon dilatation of dominant strictures is better than papillotomy and stent implantation, at least concerning side effects. This is another recent study looking at patients with dominant strictures and doing ERCP and dilatation, in particular those with dominant strictures. A scheduled ERCP approach is better than waiting for cholangitis or other complications. So what is the problem that we face in the therapy? And I think the big problem is that we do not have consensus on endpoints. This is why major drug companies are reluctant to develop therapies for PSC. There have been major meetings with EMA and FDA to find a consensus on endpoint. And as far as I understand, the consensus of these meetings is there's no consensus. And it is very important that we use these prospective trials of therapies, even those if they are coming up with a negative result, to find out prognostic scores and develop endpoints. And just one example, this is a study from the simtuzumab trials. And here MRCP was evaluated because simtuzumab was negative, but a large cohort of patients, more than 200 PSC patients prospectively over a long time. And then all arms were combined. And the result was that hepatic dysmorphy, signals of portal hypertension, and perihepatic lymph nodes, more than 10 millimeter, are significantly associated with clinical endpoints of PSC, like cirrhosis decompensation, dominant strictures, cholangitis, or cancer development. And based on these studies, a score was developed, and I think this is very, very important that we use these studies, even if they are negative, to develop data and scores that can be used for endpoints in future therapeutic trials. MRCP is costly, certainly. There may be even easier endpoints. There's a recent interesting paper from last year that even IgG levels may be used as a prognostic score and a score for treatment response. In the last part of my talk, I want to touch on autoimmune hepatitis, where I started my career with. So what's new about the pathophysiology of autoimmune hepatitis? We understand that a loss of tolerance to the hepatocyte is mediating the disease process. This can be infectious agents, this can be drugs. There is a genetic background, as we know. But what do we know recently from the studies that may have an impact on future therapies? And I come back to therapy later. But I think regulatory T cells are very interesting. They're explored in other autoimmune diseases like diabetes. And overall, in the liver supernatant, there is a decrease in IL-2. We know that IL-2 is necessary to stimulate regulatory T cells. If we have a deficiency of IL-2, T cells, regulatory T cells, go under apoptosis, and you see then the diseased liver, you have an increase of annexin 5 positive Tregs, which is a marker for apoptotic T cells. And the problem is that there are increasing, accumulating data that regulatory T cells, that the activated ones are missing in the liver. And this is data from Dr. Taubert from our institute, where he has compared the results of pediatric and adult autoimmune hepatitis patients and have looked at the IL-2 levels under treatment response. In the diagnosis, very, very few remarks. The interesting time is over identifying the molecular targets of autoantibodies. I remember my time when I went to San Diego, 87, 88. I was among 3 groups in Dr. Eng Tan's laboratory to clone the target of the LCAM1 antibody, which is the cytochrome P450 2D6. PDHE2, as I mentioned, discovered by Eric Dershin, is the target of mitochondrial antibodies. But we agree that as this is a cartoon now, which is a little bit different from the European guidelines, the American guidelines, the AASLD guidelines, are rewritten now and are in press and will be published this year. There is still a group of patients that presumably has autoimmune liver disease responding to immunosuppressive, lacking autoantibodies. And so Dr. Taubert and Dr. Jekyll, they are looking for other autoantibodies, and they come up with a strange name now, the Huntington interacting protein 1 relecting protein, which is also targeted by autoantibodies, which are more specific than ANA and SMA. What we are looking for would be a combined assay to diagnose autoimmune hepatitis with a single marker. We are not there yet. So what's new about therapies in autoimmune hepatitis? Number one, induction therapy. Induction therapy, standard of care, is steroids plus minus azacitidine, which shows to prolong survival. What is interesting is how much steroids. And this is a retrospective analysis by Dr. Pape from the Netherlands, published in Clinical Gastroenterology recently, just a year ago. 451 patients, 9 centers from 5 European countries. We were one of the centers, and looking whether more steroids is better. The answer is no, not too much. And they took 0.5 milligram per kilogram body weight. The guidelines say we should use 1 milligram per kilogram body weight. And you say that the results concerning biochemical remission, cumulative steroid dose, and specific, and is not different. So there is some hesitation to use high-dose steroids in the future. And this is another study which is also interesting, looking at adverse events related to low dose of steroids in autoimmune hepatitis. And you see that there's a dose-dependent effect on cataract, fracture, and diabetes. However, that even low dose and also the topical steroid budesonide have some side effects that are linked to the steroid use, and therefore less steroids is better. But the big problem is not the standard of care. The problem is, what are we doing with the patients who are not responding to standard of care? And here we have another problem. Autoimmune hepatitis is a rare disease, so failures to standard of disease are even a rarer disease. And we understand, I don't want to repeat this, according to guidelines, we should use mycophenolate, MMF, if there is intolerance to azacitidine, less favorable if we use it in non-response to azacitidine or steroid. Then we may use tacrolimus or cyclosporin, which is favored in humans, in children. But we should not forget, none of these second-line therapies for autoimmune hepatitis, failure to standard of care, is approved yet. And the problem is that all these molecules we are using are off patent. So who is paying the money to develop these drugs for these indications? Therefore, the focus now are biologicals, like in other autoimmune diseases, like in rheumatology, like in inflammatory bowel disease. In Germany, for example, gastroenterology and hepatology are in one unit. So we see that the hepatitis C in gastroenterology of these days is colitis and Crohn's disease. Here is where the movement is of drug development. And interesting that these drugs, TNF antibodies, B-cell antibodies, are in the focus. And I want to give some review on the data we are seeing in rituximab targeting the B lymphocytes. This is a recent, very interesting paper, which is a retrospective analysis of 22 patients from United Kingdom, Canada, and Germany, and showing that rituximab, that there are no serious adverse events, and that we see a decline in alanine aminotransferase and in IgG levels, and that we see no flares in these patients. So this is a promising approach to target B lymphocytes with biologicals, and also we can reduce steroids. This is the largest series so far. So targeting the B lymphocytes seems to be a promising approach for treatment failures to standard of care. And VAY736 is an antibody which is being developed now
by a larger pharmaceutical company. And this is a monoclonal antibody which is already approved for other indications, other autoimmune diseases. It leads to B-cell depletion by antibody-dependent cytotoxicity. In addition, it leads to BAFF receptor blockade, and therefore it may be better than rituximab, and it's now explored in a clinical trial. However, the clinical trial has problems to recruit patients. Autoimmune hepatitis is rare, failure to standard of care is rare, and here the authorities, they insisted on having very strict inclusion-exclusion criteria so that not too many patients are left. At the very end, I want to come back to regulatory T lymphocytes. As you heard, that in the pathophysiology, they play a significant role in autoimmune diseases. A lack of regulatory T lymphocytes at the site of inflammation is obviously crucial to the disease process. So we need to either bring external Tregs into the patient or to stimulate and activate the remaining ones, and their low-dose IL-2 is very interesting. So the approaches for future therapies, whether we can increase therapeutic response by strengthening immunoregulation, using either direct adaptive transfer of Tregs or low-dose interleukin-2. It has already been shown that if we give Tregs, and this is explored in other indications like diabetes and other autoimmune diseases, and when we use these in liver patients, we could not show efficacy yet, but it was shown that those patients accumulate the Tregs at their home in the liver, and there is less targeting of other organs besides spleen and bone marrow. These are good safety signals. But it would be even better if we not need to transfuse Treg lymphocytes, which would also be very costly, if we just could give low-dose interleukin-2 in refractory autoimmune hepatitis patients. And these are the first two patients in the literature. You see here that at least in one patient, we see a significant decline of AST levels to normal. And there, another company is now exploring low-dose interleukin-2 in patients with refractory autoimmune hepatitis. So at the very end, I want to stress that the problems we have is to recruit patients for these rare diseases like PBC, PSC, and autoimmune hepatitis. We don't need standard of care, but we need patients not responding to standard of care. And if we want to have investment into these rare diseases, we also have to show that if you invest in rare diseases, that after finishing your trials, you still have patients left to treat. And therefore, the European Reference Network for Rare Liver Diseases is very important. It was initiated under the leadership of Newcastle Dr. Jones, but thanks to Brexit, the leadership had to move to Hamburg, and we are extending this European Reference Network, which is very important to do prospective and also retrospective analysis in these registries. And you should keep an eye on this reference network, because this will be the soil to develop future trials. I will be very short. These take-home messages are too many. I want just to give the message that these rare autoimmune liver diseases, that they are very important for the future, that we can prevent end-stage liver disease like cirrhosis and cancer if we use the right treatment at the right time, that a lot of movement is thanks to the new molecular targets, and that we have molecules available now that we explore in the clinics, but we need large cohort of patients, and this will be necessary as an international effort. Thank you very much for your attention.
Thank you, Michael. Magistral presentation. You were born with autoimmune diseases. Okay. Thank you. This is a PHC award.
Thank you very much.